Okay. So what I figured we'd do, guys, is today we would do the population ecology. There's not very much on it. And then we'll start into the molecular genetics, okay? All right. So there is a question in the multiple choice regarding these concepts. So are you guys comfortable with the difference between exponential growth, right, and a population? You know what it means, like it's a J curve, right? It's rapid growth, right? Bacteria was the example that we used, right? Okay, you're not gonna have to worry about this. I'm not asking you this formula, but you do need to know this one. And what I ended up doing was at the very, 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 very end of the exam, I wrote down the formulas that you would need. Okay? All right, so you don't have to memorize them. You have to just make sure you understand it, right?
Like
so,
how to apply.
Yeah, exactly. So there is going to be a question which is similar to this one. Do you guys recognize this question? So we were looking at meerkats. I'll give you their R max in a year. I'll give you the original population numbers. So how you would answer this type of question, make sure you write down the formula. Show me the numbers, like plug in what you did. And then the answer here is 140, but you then have to add it back to the original population. So let's see. Okay, which is 400. Okay? Good? All right, so you're gonna see this for sure, okay? Population density, we talked about this, the difference between them. The concept of density dependent factors and density independent factors is going to be a multiple choice question as well. So, density dependent factors, this makes sense though. If you're a very close population in terms of your, like literally proximation to each other, there's going to be more competition, less space, disease can go through, we saw it with COVID, right? Food availability will be, again, connected back to competition as well, right? Density independent factors are anything to do with something that isn't specific, like for example, any type of natural disaster, hurricanes, pollution, forest fires, all of those would be examples of independent. So you guys good with that? So make sure you know the difference. Like I said, don't go crazy on it. It's a 1-2 mark question in the multiple choice, okay?
How many questions in this unit are there?
Not many, like
40 maybe?
something like that, yeah, give or take. This is also important, the concept of carrying capacity. Does someone want to just say this quickly again? What does this all mean? Okay, good, it's an S curve, that's important to remember. So over time, a population may start off going like, if it's in a new environment, but eventually it goes to its carrying capacity. Why would that be?
Can't support anymore.
Right, the environment can only support a certain amount, and it connects with density dependent factors because it's going to slow down the growth of a population, right? So we call that K. Don't worry about the R and K selected species. I didn't ask anything about that, but you are going to have to use this formula as well.
And then that's going to be back to the exam.
And this formula is there, yeah. Okay, and then here's just an example of one of the questions. So it's very similar to before. You're just going to multiply the growth rate times the current population, and then whatever the number is for the carrying capacity, you're just going to then plug it into the formula. It will give you a number. You then add that number onto the original population to give you the new one. Okay?
It's a change in population size isn't on the exam, but exponential growth is.
This is everything I just said. This is, right?
I mean, like, sorry, I missed it right at the start. There's a formula.
That's on, oh, this one isn't.
Yeah, this one.
That
It's
one
not.
isn't, no. You guys did that in geography in like grade 9, I think, anyways. Okay, I think that's it for this.
For the unit?
But it's just population.
Yep, I don't even ask this.
Oh.
Okay, so just the things I just said there. And you can watch the video and see after. Okay?
So
Yes.
nothing on abiotic and biotic.
You should just have it in the back of your mind, but it's more of like exactly what I just said. Just study those things, make sure you know the difference between them, and you should be fine. Okay?
So this isn't on the outline, but do you know just about how many questions the exam is?
I know there's 18 multiple choice.
18.
18. I think there are. I mean, it doesn't really matter. And when you guys actually get the exam, please do not panic that it is 11 pages long. It's because there are diagrams on there as well, right? Okay, and there's a calculating ATP question, so that takes up like a full page. So yeah, don't panic with that, all right? You'll have time to finish. Just, again, make sure that you look at how much the question is worth. Julie, I'm saying that more to you. Okay, you can use point form at any time. It's not. I don't really care if you write in full sentences. Just make sure you get your ideas down.
And if we're like, can we make as a general rule, whatever the question is out of is the amount of points you should make?
Roughly, or it might be if it was a 6-mark question, I might like 3 points with more of a description. I think you'll be able to tell. Like, I'm not hiding anything. I'm basically telling you exactly what you need to focus on, right? So, all right. So that was it for that one. So I want to move into molecular genetics. Any questions up to this point about anything? All right. So for molecular genetics,
Ooh, that.
the big questions that are going to be on there is there's going to be a DNA question. There's going to be a few questions about just showing your understanding of transcription and translation in the selective response. So what I figure we'll do is we'll go back through, just review everything, just to get it fresh in your mind, and then maybe what we should do is a practice question on making, if I give you the DNA strand,
Oh, transcription.
transcription, translation, and so on. Okay?
Was that this term or last term?
This was actually in term 2. And it went term 2 kind of into term 3. I'm just looking for proteins.
Four of us.
Okay, so let's just kind of, we'll talk about proteins. Oh, there's actually a slide that I want you guys to see. There's a question about it.
Yeah, because I don't have straw. No, look, it's a little bit. Yeah.
Oh, here it is, sorry. Introduction to proteins. Anyone have allergies at this time of the year? A little bit? Grass, so bad. It's starting to get bad. I can feel it. Feel that great. All right. Okay. You should be familiar with this. We've talked about this. Proteins are made of amino acids. You don't have to memorize any of the amino acids. This is an important slide. Okay, so
The dice.
that's the slide I want. This is an important slide. All right. When proteins denature, we've talked about this a number of times in a photosynthesis unit when we were looking at enzymes degrading in our proteins, right? Now, how is protein, the amino acids, bonded to each other again? What are some of the different bonds that you see? Julia?
Peptide, disulfide, hydrogen.
Okay, good. So if we look, remember between the actual amino acids, right, you have peptide bonds, which are strong bonds. Then what are these ones supposed to represent?
Disulfide bond?
The disulfide bond. Are they as strong?
No.
No. So if a protein is going to denature, is it the peptide bonds that are breaking, or is it the disulfide bonds that are breaking? Are they all breaking?
Disulfide.
It's the disulfide. So this is really important kind of concept to have in the back of your head. Why a protein works and functions is based on its shape. It's such an important thing. And if you don't have the shape being correct, then it's not going to function. We've seen that. We saw that with cystic fibrosis. We saw that with sickle cell, right? So when a protein denatures, peptide bonds are still intact, disulfide bonds are not, and so what happens is the molecule will then misshapen. Okay, so you should remember that.
Yeah, if we have a question like that, can we explain it with what we know with chemistry?
You can, but you're not gonna need to, it's a selective response question.
Oh, so
Okay, alright, that's all for that. Now, if we go to,
stuck up
Let me see. Why don't you just crush it?
It's
Let me see.
stuck. It's like
Feel it with your hand.
I don't know.
Oh,
that
it's
really-
like a familiar. Like, that's just the piece.
You hear that, right?
okay,
Red Bull familiar.
there isn't a lot on this, but you should just have this in the back of your mind, because we did do a big, this was on the test, right? So we talked about cystic fibrosis,
We talked about one
okay, and some of the different conditions with cystic fibrosis. How it's why it's not functioning. Again, it's based on the misshapen
Oh.
protein, right? And then sickle cell is the same, right? So these are just examples of situations where you have a improperly formed protein.
If
Okay.
we-
Really isn't a lot on this. Don't spend a lot of time on this, okay?
Oh, there.
Not really.
Oh,
not
no,
much.
it's not asked about.
Okay.
Multiple choice or?
If it's there, it's in the multiple choice. Yeah.
If you asked about one of them on the test, can we assume the other one is on the exam?
Not necessarily.
Okay.
Yeah, like I can't ask you everything. Right? Okay. This one, selective response, just be aware of it. Okay, so this was also on the test, right? Griffith, he was trying to find a vaccine for bacterial infections. He ended up discovering the law of transformation. Do you guys remember what that was?
Yeah, it was like dead bacteria, or like virus, dead virus, or whatever. Like,
No, that was the other one. What's this one? Look at the picture.
Yeah, like when you kill the deadly
Not virus.
Oh, back.
Yes. Julie, why?
They pass along the death trait.
Right, so it's just showing you that the DNA inside of the dead bacteria, when it was mixed with live bacteria, they took that particular gene, right? And then it killed the mouse, right?
Yeah,
Hershey Chase was the one with the viruses, right? Remember they proved that it was actually DNA that was the hereditary, not the proteins, which is what people kind of were debating at the time. And then just a few things, like you should be aware that this was the guy, this was actually a multiple choice on your test. He was the one who was looking at the bases and was able to figure out that C and G go together and A and T go together.
Right.
Rosalind Franklin was the one who took the picture of the X-ray. Remember that? And because of that, these guys stole her work off her desk, and eventually came up with the model for DNA, which we accept today, which
So
is
we
a
should
double
know both.
helix, right? Just be familiar with some of the scientists. Multiple choice.
Would you, if you asked like who discovered like that or whatever, do we say the two men or do we say that?
Well, like I said, it's a multiple choice question, so there will be a correct answer.
Yeah, so it's not asking you.
It's
Right.
the two guys.
Because it would be the two guys came up with the shape, but she's the one that did the crystallography. or
Yes,
whatever, right?
yes, but they're the ones credited with the model.
So you're gonna ask who's credited?
No,
with
I
it?
don't know. I'm just telling you. Don't.
So
I,
specific.
Yeah,
I can't
yeah,
be
just
that
the way,
specific.
The way you're explaining it.
No,
Okay.
but you can't ask like which questions
Yeah.
she's gonna ask.
No,
Okay.
I'm asking how would you answer it if it was
you should be familiar
Two guys.
with the structure of the DNA, so that the sugar and the phosphate groups are in the strand. The bases are coming off of the sugar phosphate group. There isn't anything on there where you have to identify a molecule, what it looks like. Didn't have space for that. All right, now one of the things we talked about, and there's not a lot on this either, is DNA. And hold on, let's see if this is this one. This is the structure one that I showed you guys. Let's see. Okay, DNA replication. I didn't put a lot of DNA replication on this. It is in the multiple choice section though. So what you're gonna need to know is you just wanna make sure that you know the basic process. So I want you to understand what happens. So remember how we talked about a variety of different enzymes,
Ooh, helicase.
right, that are involved, okay, in replicating DNA, right? So again, please don't spend a tremendous amount of time on this because there isn't a ton on the exam about this. So just be kind of familiar with what the enzymes do. So helicase is unwinding.
Is this,
What is this one doing here again? The type
yeah,
polymerase.
breaks back.
It prevents supercooling, which means it stops it from over
Snapping
twisting.
snap, right? Oh. Which one was the one that stops them from snapping?
That's it. Super.
Sure, yeah,
If
go with
it's
that.
super close, it's not gonna happen.
Okay, yeah, it
so
just
can I
stops.
say,
It prevents it from over curling, like so that and then it could snap back for sure. All right, now how does it work again? So if you were to be creating a new DNA strand, right? So we're creating a 3 to 5, a 5 to 3, and then a 5 to 3, 3 to 5, right?
Hi, Mr. Knight. I've come for your balls. Can I take them?
Sure, yeah,
Thank
go
you.
for it. So
Have a nice day.
you too. So how does it work again with the DNA polymerase 3?
bye, Rob.
Bye. Oh, this stuff is great, guys. You should listen. It's real cool.
He loves us. He's going
Not
into
what
this.
we weren't listening.
Okay, what's DNA polymerase 3? What is it responsible for doing again?
Sorry, which one did you say?
The third. DNA polymerase the third.
Doesn't it like replicate from in a 5 to 3?
Close.
Or in a
It reads 3 to 5, so it's going to read the leading strand, right? And puts in the appropriate bases that go with that. So it's making this 5 to 3 strand, right? Okay. Now, the only thing is, though, is that it can only read in the 3 to 5 direction. So do you remember I was saying that on the other side, the other strand is going to be read discontinuously? So you're going to have these areas, Okazaki fragments, which are just, you know, a group of the correct bases that are supposed to be there. However, there's gaps in between.
and that's for the RNA.
Now, RNA primers, remember that they're the ones that go to a certain spot on the DNA strand and it basically signals where the DNA polymerase 3. So imagine them just kind of working together, right? Remember we talked about primers, like in painting, right? Before you're going to paint a wall, oftentimes you prime it first. So it's basically the first step and it will signal where to actually start on the DNA. Final thing is, so you have your DNA polymerase 1 removes the RNA primers and adds in bases that are missing, right, on the discontinuous strand. And then the last step was the ligase. So the ligase, can you tell what's happening here again, based just on this picture?
Isn't it just filling in the gaps
Of what?
between the fragments?
Okay, you're right. How?
The phosphate and the sugar.
Right, so the phosphate and the sugar need to be bonded together actually on the strands. All right, so that's when the ligase comes in and that's what it does. Okay, it's used a lot actually in genetic engineering, right? Because what they do is if they want to break DNA, like snip it, then they want to rejoin it, they use the ligase. Okay, so just remember ligase makes the sugar and phosphates of the strand bond back together again.
Is this all multiple choice?
Yes.
Oh,
Yeah.
this is all.
Yeah.
There's no short answer.
Not on this,
Or
no.
long answer on this.
Not on this in particular, no.
It's like thank goodness.
Yes, there's so much. Right? I can see why you're wrong. Okay.
So just know
Now,
what the
then
enzymes
we
do.
got into, this is where there's going to be more marks, right? Is the transcription and the translation. So let's just review just how it works. Then we'll do a practice question on the board together, just to make sure you guys know how to do a question like that. Because you are going to be given a question where I'm giving you, you know, the strands or something, and you're going to have to like just, you know, make tRNA molecules and mRNA and all that fun stuff. So, okay, so let's go to transcription first. You guys good with the way I'm doing this?
I think so. Are you? City. You sure? 100% sure.
Okay. So first of all, you should be familiar with this back of your mind. You know, it'll help you when you're doing, you know, the overall big problem as well. If RNA is just a single strand, DNA is double stranded. What's another way that you can tell the difference between RNA and DNA?
The basis is different.
Okay, do you remember which one?
Uracil, thymine.
Good. So thymine is found in DNA only. Uracil is found in mRNA only. That's an important one to remember because that's going to apply to that big question, right?
Will we have to explain what we're doing for a question like that?
No. It's literally just going to be you drawing everything out and showing you that. Okay, now, if we're looking at how, so remember that transcription is taking place inside of the nucleus, and you're creating the mRNA. So remember the DNA doesn't leave the nucleus, it's going to stay in there and it creates a message. So this is showing how we create the message. This is called the transcription, right? So you have an initiation where it attaches to the strands. We're looking at RNA polymerase this time. Now remember, it's attaching to the 3 to 5 DNA strand and makes a 5 to 3 mRNA complementary strand. It's not the copy. Okay? But there's tricks, you know. Like you could look at what the other strand is, the 5 to 3, and go, oh, I'm just going to replace all the T's with U's, and then I have it. Right? There's faster ways to do things, right? If you know when you're doing an actual question. So it basically creates a strand. It does a complementary basis, and then when you get to a stop codon, it terminates. All right. We talked about how in order to make sure when it actually leaves the nucleus, it needs to have the 5' cap and the poly A tail so it doesn't get destroyed in there. And then we talked about this a couple of different times, that in reality, the way it works is pretty cool. You can have a pre-mRNA strand, which depending on how you either remember the exons go out, introns stay in, and you can mix them up. So you could potentially have multiple genes coded for one mRNA strand. Okay, any questions about transcription?
We don't do the splicing. We never did that, right?
What do you mean the splicing?
We never we need to understand that, but we don't
Understand it, yeah.
We don't do that. Right?
No, not in the question, the big question that you did before.
The DNA you transcribe it always has to be 3'5'.
Yes,
So is that strand technically?
but you can go the opposite way, right? There's a leading, just for now, it's more complicated than that. Just understand leading strand, lagging strand. You want to go to the leading strand, which is a 3 to 5.
Is this? Oh, I'm sorry.
Did you have a question, Julia? Sorry, I thought you said
Oh,
your hand up.
yeah, so we don't need to know like post-transcriptional control factors?
No, you do. You will. This is just transcription. That's genetic control, which we're going to get into. There is a question on that.
This is just like technically when we're doing that like for the big question when we're making the mRNA, is that technically pre-mRNA?
Technically it is, but in the question, you don't have to draw in the 5' or if you want to, you can. It's for fun. But it's not mandatory, so. Okay, translation. So now we've left the nucleus. The mRNA strand, where does it go?
We left the nucleus?
It's going to go to a ribosome, right? So here's just an overview of what happens. So here we've transcribed it to an mRNA, and now we're going to make the mRNA is going to now code for certain proteins. This is hopefully pretty familiar. I don't feel like we did this that long ago.
Yeah.
Right? All right.
This is the one
What's
you
the
had to test
purpose
on.
of tRNA molecules again?
Create the amino acids.
They don't create. They create.
Get them.
They bring them, right? So remember that they're like T stands for transfer. They move around. They go into the cell. They find the appropriate amino acid. It's not just random. They actually are very, very specific. And what makes them specific is they have two things. They have the anticodon, and that is going to attach to the mRNA strand, which is on the ribosome, right? And then the amino acid that they're getting, right, is on the codon end. So you should be familiar with this. On the big question, I don't care what your tRNA molecule looks like, just make sure that you indicate, though, which is the codon end and which is the anticodon end, so that I know that you know.
So doesn't matter if the shape's all.
Yeah, like I draw mine like this, like almost like the Riddler, you know?
Yeah, yeah.
Good question.
Right, but and yeah, there's different versions of them, so I'm fine with just however you want to draw it. Just make sure you clarify that.
Like we can draw whatever we want.
Yeah.
If you do like a line.
Like a stick man.
If you must. Just make sure you clearly label the anticodon
I'm
and
gonna draw
codon.
my
Okay. Ribosomes. Remember that there are two subunits. The ribosome moves along as the mRNA is being read 5 to 3, so in the direction that you're going to probably draw it in, because we all kind of draw this like we write this way. It's usually what happens. There's 3 sites Okay, where does the initiation all begin?
P.
At the P site, good. So the P site is where your first, the AUG, right, which is a start codon, that tRNA comes in there, then the whole process begins. So if we look here, this is just an overview of everything. There's basically three parts. There's the initiation, elongation, and termination. Initiation is when you actually start the whole process, right? So here is initiation. Like Mitchell said, starts on the P site. Now we're elongating the molecule, and what are all the numbers? 1, 2, 3, 4. What does that all mean again? Like these little circles. What are those? Those are the amino acids that are being bonded together. So the little dark line in here is supposed to represent them being bonded. Remember that every time a tRNA molecule comes in, it's going to the A site, and then whatever is on the P site is now going to attach to that, and then everything moves down one. The one that's leaving goes to the E, the one that was on the A goes to the P, and then the new one comes into the A. Does that sound familiar? Again, just make sure you have this in the back of your mind because it will help you when you're doing that question. You don't have to draw all of this because that would be very difficult. You'd be quite an artist if you could do that. When we get to the end,
Sorry,
Yeah.
we're not gonna have to like write anything about this. Maybe.
just it makes it easier for you to do if you just have it in the back of your mind on how these things work, right?
Yeah.
Because you may be able to add it into questions sometimes if you want to try to give an example of something and that's in your mind, right? Termination, how do we know when to stop?
Stop codon.
There's a stop codon, right, on the actual mRNA, which then is going to signal a bunch of other things to happen, and basically you get a release factor, things breaks apart, now we have this beautiful protein, okay?
Are you gonna give us a notation on that?
Not necessarily, maybe, we'll see, right? You're gonna have something that looks like this on the exam, because you are gonna have to be able to code the proteins, right? So does everyone remember how to use this? Okay, it's pretty straightforward, right? If I wanted to do, what is CUU? What would that be? CUU. Correct. Stefan, you're good?
Yeah.
Okay.
Can I have the wash?
Yeah, of course you can. All right, so why don't we do the big question now on the board just to see if you can apply all of this, okay? We'll just pause there for a second.
Do you go in class?
Yeah.
Can Mitchell try and get everyone to Mitchell always
No, no, like we're leaving.
Oh, you know, leave blocks. It's just maybe the guy is supposed to have a presentation. So,
Okay, this is just completely random, so hopefully I didn't hit a stop codon or anything, okay? All right, so let's say this is our DNA strand. What would be the, this is our leading strand, because I've drawn it from a 3 to 5, right? What would be the complementary DNA strand?
so maybe
Okay, go ahead.
A, T, okay, is that G,
G, yep,
C, C, T,
good.
and then C, G, T.
Good.
You have T.
Okay, good. So those are your two DNA strands, right?
Oh, this is replicate. Okay.
No. This is transcription translation. You're just right now. You're looking at the 3 to 5 and the 5 to 3 DNA
Oh,
strand.
that's a DNA.
Okay, that's all DNA. Now, I want you to make the mRNA strand.
Now.
So Julia, can you make our mRNA strand for us?
Sure. Sorry, T.
Okay, hold on.
I messed up. Sorry, U,
Okay, we're going with this one.
A. Okay.
Okay, so this is the one you want to code. You're doing the complement to this one.
Okay.
So you literally you could use this one, but anytime you see a T, you replace it with a U. Okay.
So then A, U, C, G, G, U.
Okay, be careful.
C. Sorry, C, C, U.
Yep.
And then C, C,
CG.
U. Oh, C, G, U. Sorry, I thought that was.
Good. Okay. Or course, now look,
Sure
just,
you didn't.
so,
Sure.
yeah, if you don't like, and that's okay, I don't like doing it that way either, to be really honest. I like to just do the opposites, cause that's always how I work. So notice how it's exactly the complement. AUC, CCU, CGU, complement to that one.
Shouldn't it be AUG?
Yep. Okay, everyone good with that?
Well, wait, what's the method that you don't like?
No, not that I don't like. It's just I like to go opposite all the time. But the other technique would have been if you have the 5 to 3 DNA strand, you could have copied it exactly and just put in U's where there are T's.
Okay, but you like to just do it.
I'm just saying what I like, right?
going on.
Okay, but you can do whatever you want. It's your exam, right? Okay.
What's my example?
Next thing I want you to do is I want you to draw the tRNA molecules. So this is how I would do it. Just again, I would probably draw them right under the mRNA strand, just to make it so that it's easier for you to see, right? So if it's AUG, this is going to be my anticodon N. What would be on here?
Do we also label tRNA molecule?
If you say anticodon and codon, I'll know, right? This is the tRNA. So what's my anticodon N here?
UAC.
Okay, and then what would be over here?
AUG.
Right. And then what about this one?
GGA.
And down here?
CCU.
Good. See the pattern? Okay, last one.
GCA.
And then down here?
Only because
CGU.
5 months.
Okay, so there's your, now notice how I am going like this. Or you can, even if you would like Mitchell, you can do this as well. Okay, just make sure you're really specific so I know that you know.
So I have a question about, so this is about like the way I wrote it for my last test.
Yeah.
It was like, so I drew a T, right? And you know, like at the end, you know how there's a circle? What I did was I put the codons there, and then underneath it, I drew the amino acids and I linked them.
That's fine. Okay. So now that what you would do is you would go to that sheet. You can draw something like this if you want to show that your representation of the protein. So if it's AUG, this was actually our start. Doesn't necessarily, I'm trying to remember, I think on the exam, I actually have a start and a stop codon in the molecule, okay? What would CCU be again? I don't know if you guys, I'll just do it here. C.
Oh, we also know the mutations, right?
I think it's Foley.
Here's CCU. CCU is proline, that's very good.
On the test, the one thing I missed was a word.
What's that?
On the test, I got all of the codons right, and then for some reason I just started reading the chart like completely wrong.
Okay, but you know how to do it now,
Yeah.
right? Okay, good. Yeah, that's good. Okay, and then what would CGU be?
Alanine. Wait, no, never mind.
Arginine, right? Notice how I'm doing these, because this is what we used to do. Like we always just put the first 3 letters. You can write it the whole thing. You can put the first 3 letters in there. That's fine.
And then make sure that they're connected.
Yep. Okay, how's that? You feel comfortable with this?
Yep.
All right, good. Okay.
Well, we also need to know all the mutations, right?
Okay, so that's going to be our next thing. We're going to talk about mutations. How are we doing with that? Oh, I'll have time. That's good.
Is there gonna be a question? It was the test we wrote in December. It was like a whole chart. I forgot what it was about. It was a whole chart.
Oh, that was a calculating ATPs.
Oh, yeah. Did you get that question?
There is.
That was.
I wasn't
remember-
here.
Oh,
That's why.
I remember that was easy, but it was like. a lot of-
You know what? You just have to know that summary sheet.
Yeah, no, because I remember it was easy. But it was
Yeah. Did you end up getting it? Because you said you wanted it.
Oh no, sorry, I forgot to ask you.
Okay. All right. Yeah. I'll send it. I'll send it to you at the end of class.
Forgot you
actually.
weren't here all the time. That was like.
This guy, should we know the like steps of the summary sheet?
So it's literally it's going to be a very similar question to what it was on that test. So you're going to have to write out everything. So, and I'm going to tell you, like for proteins, I'll tell you where they enter. You kind of have to, yeah, you should know the steps.
But
Okay.
also, what about like fatty acid metabolism?
You definitely know, there's definitely a fatty acid one. Maybe what we'll do is on Wednesday, we'll do one again just to make sure everybody knows how to do it. All right, mutations.
Yeah.
It's in here.
I've never been in my phone.
Okay, this isn't too hard. You're not gonna have to know the one from last year, the chromosome mutation. You just need to know the gene mutations because that's what we focused on, right? Okay, so what would be a silent mutation again?
Or
Just
when
look at
it
the
doesn't
picture.
change.
What do you see?
Oh, like it's mutated, but it's still the same.
Okay, now when we say, can you just be a little more specific? What's being mutated here specifically?
Oh, that's fine.
So we're changing a letter. All right, we're not inserting or deleting anything. We're actually literally a spontaneous change of a letter, right? In this case here, it doesn't matter because they end up being the same anyways, right? It codes for the same protein, so dodged a bullet. That one. Okay, next one. What's missense?
Wrong amino acid.
Okay, so you now have another change in the actual letter, which is going to code for the wrong amino acid. Good. Then what about nonsense? Everyone's
Stop
favorite.
code, a premature stop.
Okay, good.
Changing the letter which codes for a stop.
Correct. Good, good. So that can happen like in the middle of it, at the beginning. It really messes everything up,
But I have a
right?
question though. What if there's a mutation? I feel like we've definitely talked about this in class. I just forget. Where there's a mutation and so there's no stop code.
I mean, it must exist. It's not a common one that has, like when I research this, I don't really talk about that, but I'm sure that could be a possibility.
Like I know it's still like one of the whatever, but is it like what happens to the actual gene?
If there's anything wrong with the code and it causes the protein basically would not form properly and then it wouldn't necessarily function, right?
Oh, so then it would
Yeah,
just
it's
be
kind of
like-
that's kind of a general thing that always, kind of is always the answer to that. All right, here we're inserting, okay, an extra letter. And then what's the overall effect if you insert the letter, another letter in there?
Shifts everything down.
Right, so it's going to shift everything down, which means wherever you began, you've changed the order in which they code. Remember, they're coding 3 at a time. Okay, so you're going to get different amino acids, right? And then the last one was the one where we had a deletion. Same idea. If you take one out, it's going to affect anything after that. And it's going to now have a different thing. Now, keep in mind, and if you guys remember, during our lab, remember that mutation lab where you guys were building the mutations? We did have a scenario where you had 2 of them. Like, technically, you could have a scenario where you could have an insertion, right, that causes a nonsense. You could have, probably the only example, I'm trying to think if you could have another one.
But what about a deletion and then an insertion?
If you had an insertion and a deletion, and it was a silent mutation, right? So you could potentially have, that can get it really, really complicated. So just keep that in the back of your mind that sometimes there can be 2, okay?
What if there's like multiple deletions so that's non-friendship?
Well, then it probably, I mean, like I said, you can have multiple things going on. Like you could have more than one deletion or more than one, right? Doesn't just have to be one, but it would still be considered an insertion or deletion. My allergies are so bad, sorry. I was outside and the wind was blowing and now I'm all messed up. Okay, I think that's all we have time for today. All right, does anyone have any questions about anything we talked about? All right, awesome. I'm gonna put something on my YouTube channel.